All right, so last time we were talking about what name reactions? Claisen. And what is the Claisen? All right, so that's right. So it's the reaction of two esters to form a beta-keto ester under what conditions? Thermodynamic is part of it, that's true, but was it under acidic or basic conditions? Technically it could be, but what were we talking about? I could show a mechanism under acidic conditions, but it's under basic conditions. That's right. And so the Claisen reaction gave us beta-keto esters, right, from the reaction of two esters. And we also had a crossed Claisen. What was a crossed Claisen? Two different kinds of esters. What are the limitations of the crossed Claisen? You can get multiple products, up to 4, not counting enantiomers, right? And what else, Gabby? Oh, that's what you said, okay. That's right. We need one ester that doesn't have alpha hydrogens for it to be practical, right? Now, we also talked about some of those things. There was also a reaction that formed a 6-membered ring that was like the Claisen. What's the name of that one? The Diekmann, right? And so it's just a Claisen that happens to have the two esters tethered together, right? And so it can form 6-membered rings. We can form 5-membered rings, depending on how many carbons we have between the two esters, right? And then we started talking about this reaction that we see here. What's the name of this one? Malonic ester synthesis, that's right. So I think this is where we ended off, right? So we generate the enolate. Enolate then reacts with an alkylating agent. In this case, I picked this one. We form a new carbon-carbon bond. And then when we heat it up in the presence of water and acid, what happens? Describe for me what happens. Ultimately, that's what happens. That's right. A little more detail. What happens to the esters under acidic conditions when I boil it up with water? Yeah, I hydrolyze the esters to a carboxylic acid, and that diacid then undergoes a decarboxylation to give me the carboxylic acid. So we lose carbon dioxide in the process of this last step that you see here. Okay, so let's continue on. What product would you expect from this reaction sequence? What do you think, Chance? Yeah, you need to draw it out probably, huh? What's going to happen in that first step, that first arrow? What are we going to generate? What do you think we're going to generate, Delaney, in that first step? Just that first arrow. I'm going to take my dimethylmalonate and I'm going to treat it with sodium methoxide. What's going to happen? What is sodium methoxide? It's a base. What does it react with? What's it going to remove? Karina. It's going to remove one of these hydrogens. Yeah, why is it going to remove one of those hydrogens? because they're acidic, right? So we're going to get an enolate. What's going to happen in the second step? I'm going to make a carbon-carbon bond, aren't I? Okay? And then when I add sodium methoxide again, what's going to happen? Huh?
So basically what you're doing is you're taking out two hydrogens.
Ultimately?
Yeah, and adding carbon bonds.
How many carbon bonds am I going to make?
Well, there's only one more.
Okay, so what's the answer? A, B, C, or D? A is the correct answer. Let's walk through it. We start off with dimethylmalonate. There you go. And when we treat this with sodium methoxide, we get drawing one hydrogen, we get the negative charge there, right? Now what happens, Matthew? Yep. Now what happens in this step? All right, that's right. That's ultimately what happens in the second step. That happens, right? And that's how you get the product. And it forms a cyclopropane ring, right? So we can form 3-membered rings, 4-membered rings, 5-membered rings with this type of a reaction. Now, you'll notice that answer C shows a Williamson ether synthesis where the sodium methoxide can come in and just displace in an SN2 reaction and give product C. And I've taught you that alkyl halides plus methoxides, or alkoxides, will do the Williamson ether synthesis, right? Why does this process occur and not the Williamson ether synthesis? What's the difference? If we go through a Williamson ether synthesis versus this pathway, what's the difference? Well, a carbocation would imply that it's going through a SN1 reaction, which Williamson ether can do, but this would be an SN2 one. So no carbocation.
I'm going to guess to say because Williamson ether doesn't involve the
Not far from the correct answer. You've got a base that's fairly strong, and you've got a compound that's relatively acidic. What are the fastest reactions that we know of? Acid-base reactions. Okay? Nucleophilic reactions are slower than acid-base reactions. So when this base reacts, it's going to react as a base and pluck off the acidic hydrogen. That occurs very, very quickly. And in doing so, we generate this enolate, which then functions as the nucleophile. So in reality, you never have methoxide around enough of it that can do a Williamson ether synthesis because that reaction is slow compared to the acid-base chemistry that we're doing. Okay? So there was the possibility if you had picked C, that's not an irrational choice. You just have to take the acid-base chemistry into account. Okay? How about question 18? If we have this steroid, tetracarboxylic acid, and we heat it, which carboxyl group would readily be lost as carbon dioxide? You would say 2? Okay, and you're right. Because this carboxylic acid can form that 6-membered ring, hydrogen bound to this ketone, and we can push the arrows to lose carbon dioxide. We can't do that for any of these. Okay? So very similar to the malonic ester synthesis after we boil it up in acid. You have that beta relationship between the two carbonyls. And so that's why you get the loss of the carboxylic acid at position 2, not at position 5, 13, or 15. Those are stable. They'll stick around. But the one at 2 is readily lost. What is the product of this reaction? I've got one vote. Anybody want to take a guess? Anybody else? Oops, I didn't know where. There we go. It is B. Describe that reaction for me. How is that working? You picked it, Sam. What do you think?
So you lose an alpha hydrogen off of the compound on the left.
On the left?
On the right, I'm sorry. And
Your other left.
yeah, the left.
Okay.
The one on the right, because the one on the left is ester.
Mm-hmm.
And,
So what do we know about the pKa difference between alpha hydrogens on a ketone and alpha hydrogens on an ester?
they are different.
By about how much?
Uh,
What's the typical pKa of
it's
a
like
few orders of magnitude for sure. A typical alpha hydrogen for an ester is about where? A little higher than that. About 25. Now where's the ketone? 19 to 20, right? So again, it's all about acids and bases, right? So we've got a base, sodium methoxide. It's going to abstract the most acidic hydrogen, which is the alpha hydrogen on the ketone, okay? So we generate the enolate here, and that then attacks the carbonyl of the ester, goes through that tetrahedral intermediate, right, which then collapses down and gives us the, how would you describe that product? In the Claisen reaction, we get a beta keto ester. How would you describe this product? It is, but we have to add one more descriptor to it. Is it alpha? It's a beta diketone. That's right. So you start with one ketone, alpha, beta. So it's a beta diketone. What's functioning as the nucleophile? The enolate of the ketone. And what's functioning as the electrophile? The carbonyl of the ester. That's right. Okay. This is not a Claisen reaction. It is not a Diekmann reaction. But lo and behold, the mechanisms are very similar, right? So all of these reactions that I'm showing that came off of old retired ACS standardized exams, you really know everything you need to know to get these questions correct. Okay? Now we need to talk about the Aldol reaction. The Aldol reaction is between two molecules of aldehydes or ketones, and they react with one another to give beta hydroxycarbonyl compounds. Okay? So for example, if we start with acetaldehyde, okay, and we treat this with base like hydroxide in the presence of water, we will get this beta hydroxyaldehyde. Can anybody take a guess as to what the mechanism is? What do you think, Rose? Can you describe the mechanism for me? What do you think, Mason? What's the mechanism going to look like for this? We're starting with 2 molecules of aldehyde, right? We're forming a new carbon-carbon bond. What's that very similar to that we just finished talking about? Except it was with esters. It's very similar to the Claisen in a lot of respects, right? So what do you think the first step is going to be? That's right. The base is going to react with the alpha hydrogen. You're always going to have this acid-base reaction. Always start there. You're going to generate the enolate of one of the aldehydes. Are we going to generate a little bit of enolate or a lot of enolate? A little bit. And why do you come to that conclusion? Well, they will react with each other. That's true. But why do we only generate a small amount of enolate? You should be seeing a recurring theme. Well, that's true. pKa. It all comes back to GenChem, right? What's the pKa of hydroxide or the water? It's conjugate acid. About 16. What's the pKa of the alpha hydrogens of an aldehyde? 18, 19, somewhere in that ballpark, right? So it's not going to lie to the right, is it? It's going to lie to the left. But once we generate a little bit of enolate, it's in a large sea of reactive aldehyde. So you generate a little bit of enolate. Right? And this now reacts with the remaining aldehyde that we have that's not an enolate. And so we form a new carbon-carbon bond and an alcohol. Because we're adding a carbon nucleophile to an aldehyde. That's just like the Grignard reactions, right? When we added a Grignard to an aldehyde, we got what kind of alcohol? We got secondary. We don't have a Grignard here, but we have a carbon nucleophile, which is just like a Grignard in a lot of respects. And so that's going to add to the aldehyde to give us secondary alcohol. Okay? Again, nothing particularly new and surprising. Okay? But this is called the Aldol reaction. How do you remember the Aldol reaction? Al comes from what? Aldehyde, and all comes from alcohol. So I know my product is going to have an aldehyde and an alcohol. That's how I always remember it. Technically, this will work with ketones, and we still call it the Aldol reaction for whatever reason. Okay, but what would we get in if we had, let's say, 2 molecules of acetone reacting, what would the product be? It wouldn't be an aldehyde alcohol. What would it be? An aldehyde, or excuse me, an alcohol and a ketone. And where would the alcohol be? On the beta carbon. That's right. Okay. The mechanism, again, very straightforward. You end up generating a little bit of enolate. That then reacts with the remaining aldehyde. That alkoxide intermediate cannot collapse. All right? And so it reacts with the water that you have present to form the beta hydroxyaldehyde. For those of you studying biology and for those of you who are going to study higher level biochemistry, you will hear a term at some point about aldolases. These are biological enzymes that basically perform the Aldol reaction. Notice the Aldol reaction is occurring in water. We are mostly water. That's why we can't do Grignard chemistry in biological systems, but we can do Aldol chemistry. So we can make carbon-carbon bonds using Aldol reactions very, very easily. You have enzymes that do this. A lot of biological organisms have enzymes that do this. But in biochemistry, you're going to learn that the primary use of those enzymes isn't to make carbon-carbon bonds, but to actually break carbon-carbon bonds. Because it turns out that sugars are like aldol products. And your enzymes take those sugars and transform them ultimately into ATP. And you have to have an aldolase enzyme to help do that. Okay? And so it actually does the reverse of the aldol reaction. And we know that catalysts catalyze both the forward reaction and the reverse reaction. So if you give a biological system plenty of sugar, it will do the reverse reaction and do the retroaldol reaction to make the molecules that are necessary to build ATP. Okay? But I can also hijack that enzyme and take it into the laboratory and do the aldol reaction. We do that in my lab quite a bit. It just depends on how you play with Le Chatelier's principle, right? And so we can actually use aldolase enzymes to also make carbon-carbon bonds, as well as break them down. Okay? If we have propanal, okay, we have the alpha hydrogen. It will react just like we did with acetaldehyde. We get the beta hydroxyaldehyde, except here we have another issue. How many isomers of that molecule can we get? What's the maximum number of stereoisomers we can get for that aldol reaction? Everything. 2 stereocenters, so what's the formula? 2 to the n, right? Where n is the number of stereocenters, so we can have a total number of 4 stereoisomers. 2 diastereomers, and each diastereomer has an enantiomer. So is that going to be an efficient reaction to run in the laboratory? No. Okay. So each stereocenter can either be R or S, right? So I could have R, R, I could have RS. Now how are those related to one another? RR versus RS. Are they diastereomers or enantiomers? This center is R, this one is S, this one is R, that one is R. Those 2 products, would they be mirror images of one another? No, they are not. Those would be diastereomers. And then those diastereomers each would have an enantiomer, right? So we would end up with a maximum number of 4 stereoisomers that we could have for the aldol reaction. Okay? Now, the aldol reaction doesn't always give us as the final product the beta-hydroxycarbonyl compound. And this is where life gets a little tricky. If you're not careful, the beta-hydroxyaldehyde or ketone, whatever the case may be, can lose water, one of the hydrogens from the alpha carbon and the OH from the beta carbon, and give me a new carbon-carbon double bond. Why is that favorable to do that? In fact, I'll tell you that to isolate the beta-hydroxyaldehyde, you have to be a particularly good chemist. In the workup, you have to be very cautious because it will rapidly lose water to give you the alpha-beta unsaturated aldehyde, meaning that that must be more stable than the beta-hydroxy compound, right? So why is that more stable than the beta-hydroxyaldehyde? Conjugation and resonance. That's exactly right. Now, in this case with the acetaldehyde, this one's not too hard to isolate right here. But if it comes into contact with excess base or even a little acid, it will rapidly eliminate to give me a carbon-carbon double bond. What other reactions have we learned about this semester that make carbon-carbon double bonds? Dehydrations, but we kind of covered that mostly last semester. But you're right, dehydrations will work, and that's exactly what this is. Whose named reaction? Pardon? Wittig. That's exactly right. So this does kind of like what the Wittig reaction does. It allows us to make a carbon-carbon double bond from a aldehyde, right? The difference is that to make a carbon-carbon double bond using the Wittig reaction, you have to have an aldehyde or a ketone plus a ylide, right? Here, we're just using 2 molecules of the aldehyde to do this. Example 2, acetophenone, your good friend, you've used it a lot in lab, right? If you take this and treat it with base, you will not isolate this at all. You won't ever detect it, actually. We know it's an intermediate. We can detect it in the NMR if we're clever with our experiment. But what you will isolate is what's known as a chalcone. Okay? This is an alpha-beta unsaturated ketone. Why is this one so much more favored to do the elimination than the one at the top? I told you the top example, we could isolate the beta-hydroxyaldehyde if we're really careful. I'm telling you that in the second example, you're not going to be able to isolate that beta-hydroxy compound at all under these conditions. Why? What do you think, Carl? Why does that one so readily eliminate water and give me the alpha-beta unsaturated ketone?
It's very stable.
And why?
Because the rings and...
Okay, so the rings also give me additional resonance, right?
Yes, sir.
So I have a lot of resonance structures that I can draw for this. So like Carl said, this one is so much more stable than the one at the top. I could draw a lot more resonance structures for it, right? So I can't isolate this. It gets formed, but immediately loses water to get all that extra resonance stabilization. Okay? So the aldol reaction can give me 2 possible products. It can give me beta-hydroxycarbonyl compounds or alpha-beta unsaturated carbonyl compounds. Okay? So we can get alpha-beta unsaturated carbonyl compounds like I'm showing on this slide, or we can get the beta-hydroxy carbonyl compounds as I've shown on the previous slides. Okay? Now the mechanism for this is kind of interesting. It goes through an elimination that's a little strange. Okay? Notice what's kind of strange about this. If you look at this elimination step, what's kind of strange about it? What are you talking about? Another arrow.
Well, I'm kind of blind, so...
Okay. So this is an OH minus. It's coming in and reacting with this acidic hydrogen and giving me this carbanion, which then eliminates. What's kind of strange about that? All right, very good. Why would it form the carbanion? The 2 types of elimination mechanisms that we've talked about are E1, which goes through what kind of intermediate? Carbocation, which this ain't, right? And E2, where the base is coming in and the leaving group is leaving and the double bond is forming all in one step. Is that happening here? No. This is called an E1CB mechanism. Stands for elimination unimolecular conjugate base. Why is this an E1 mechanism? Why would it be called an E1 mechanism? Exactly. It's not all happening in a concerted fashion, right? And so what's happening is the hydroxide reacts with the acidic proton to give us the conjugate base, kind of like an enolate, right? And then slowly that eliminates to give the new pi bond. So this is the fast step, okay? And this is the slow step. Can you explain to me why the first step is fast and the second step is slow? Repeat that, please. They're quite acidic, that's right. And so that reaction we expect to occur fairly quickly. So why then does the second step occur so slowly? Well, that's certainly true. Yeah, that's one way to look at it. What's another way to look at it? So you're saying you're sacrificing a stable sigma bond and gaining a weaker pi bond. Okay, that makes some sense. But we're also gaining resonance stabilization, right? So that'll counter your argument just a little bit. So what's the other odd thing? What's the leaving group? Pardon? In the second step, what's the leaving group? Hydroxide. Is hydroxide a good leaving group? No. It is not a good leaving group. Okay? And since we're generating hydroxide as the leaving group, how much base do you think I need to have around for this elimination to take place? Not much. It's catalytic, right? And that's why you have to be really careful. So I can actually make that product fairly easily, that beta hydroxyaldehyde from acetaldehyde. But if I have just a little bit of base around, it will slowly eliminate water through the E1CB mechanism, because I don't need a full equivalent of base for this to go. It's catalytic. Okay? So we can have a reaction occurring here that's a little odd. So now you have three mechanisms to think about when you think about elimination. E1, which goes through a what intermediate? Carbocation. E2, no intermediate, it's concerted. And then E1CB. E1CB works when you have an acidic compound reacting with a base and you have a poor leaving group. Okay? Hydroxide is a poor leaving group. And so that's why I can generate that anion intermediate, which is not a carbocation, right? And then it eliminates to give the alkene. So this is known as an E1CB mechanism. I don't know if they actually talk about that in your book, but nonetheless, that's what it is called, E1CB. And it stands for elimination, unimolecular, and CB stands for conjugate base. That's right. So that's how I know I'm going to get an anion as the intermediate. Okay? So in the remaining time that we have today, we're going to talk about two new reactions. We're going to talk about the Michael reaction, and we're going to talk about Robinson annulations. I'm throwing a lot of names at you here at the end, but you already know all your mechanisms. There's nothing new for me to teach you in terms of mechanisms. Right now, we're just learning new reactions that basically use all the mechanisms that we've learned. Okay? Now, if we think about an alpha-beta unsaturated carbonyl compound, here I've drawn an alpha-beta unsaturated ketone, which we can get from an Aldol reaction, right? These molecules have special chemistry. If you're a nucleophile and you're going to react with this ketone, where would you want to react? All right, you would predict that you would attack the carbon of the carbonyl. That's what all up to this point nucleophiles have attacked when reacting with ketones, right? The carbon of the carbonyl, because we're going to break that weak pi bond, right? But now I've got pi bonds that are actually in resonance with one another. Is the carbonyl actually an isolated functional group right now? No, it's in resonance with an alkene, right? And so if I look at the resonance structures, I can draw a resonance structure where I break the weak pi bond of the carbonyl and put the negative charge on oxygen and put a carbocation on the carbon of the carbonyl. But that is in resonance with the alkene, right? And so now I can draw another resonance structure where I put that positive charge on that N carbon of the alkene. So now, Matthew, thinking about it, if you're a nucleophile, there's two places you could react. You could react at the carbon of the carbonyl, or you could react where? At that N carbon, right, that I have a positive charge drawn on this resonance structure. Okay? I call this carbon of the carbonyl the Michael carbon. It's actually the beta carbon and then alpha beta unsaturated. But a lot of organic chemists call it the Michael carbon because Michael chemistry will occur there, meaning that a nucleophile attacks that carbon, not the carbon of the carbonyl. Okay? Does that make sense to everybody? Because we can draw these resonance structures, now we can see that there are two places in these systems where a nucleophile could attack, right? All right. So let's look at an example. Let's suppose we take an alpha beta unsaturated aldehyde or ketone. It doesn't really specify here. We could have a nucleophile which will attack the carbon of the carbonyl and give me an alcohol. What kind of alcohol is that? Let's say it's an aldehyde. What kind is it? If that's an H. It'd be a secondary, but it's not just a typical secondary, right? It's on a specific carbon. This carbon's attached to the alkene. What's this carbon called? Nope. It's the allylic carbon. These are allylic alcohols. Okay? This is known as simple addition or 1,2 addition. The nucleophile attacks the carbon of the carbonyl. Nothing new. You've learned that before. But you can also imagine the nucleophile attacking the Michael carbon in what's called 1,4 addition or conjugate addition, which we've talked about before when we've talked about addition of like HBr across a conjugated system, right? And so you can go through these conjugate additions, which give you these enol products, which tautomerize back to the ketone or the aldehyde. And so we can imagine adding the nucleophile at the Michael carbon, leaving the ketone alone or the aldehyde alone, or we can imagine the nucleophile attacking the carbon of the carbonyl, leaving the alkene alone and changing the carbonyl to an alcohol. What's going to determine whether or not we go through 1,2 addition or 1,4 addition? It's kind of like kinetics and thermodynamics. It's going to depend on reaction conditions, temperature. It's going to depend on the nucleophile. Certain nucleophiles prefer to do simple addition and give me alcohols, and other nucleophiles prefer to give me Michael addition. The cool thing about it is you don't have to memorize a whole lot here. Most nucleophiles do Michael addition with a couple of exceptions. So for those of you who read ahead, what nucleophiles typically do 1,2 addition? If you have those memorized, then you know everything else is going to be Michael acceptors or Michael nucleophiles. Let's look at it. Let's do an experiment. Let's say that Jamie takes 3-pentene-2-one, which is an alpha beta unsaturated ketone, and she throws into it a Grignard as the nucleophile. Okay, so she takes this methyl magnesium bromide, she adds it to the reaction mixture, that she works it up, she isolates the product, she takes it, puts it in an NMR tube and gets an NMR. What she finds in that NMR analysis is that 72% of the product is the alcohol as a result of 1,2 addition, and only 20% is this 1,4 addition product. She can measure those peaks in the NMR and determine the ratios. Okay? So what does that tell us about Grignards? They prefer to do simple addition. That's right. Grignard reagents will attack the carbon of the carbonyl the majority of the time. Okay? Now, Gabby comes in and takes an alpha beta unsaturated ketone, and she uses an organocuprate, which is an organometallic reagent. Actually, they're made from Grignards by simply adding copper metal, copper ions. Okay? And what does she recognize? She goes and runs the NMR. What does she see? And solely 1,4, right? No simple addition has occurred. Grignard reagents and lithium reagents, like alkyl lithiums, prefer to do simple addition. Organocuprates do Michael addition. and everything else that's a nucleophile that you all know about also does Michael addition. So what do you need to know? If I give you an alpha beta unsaturated ketone and a Grignard, what are you going to tell me the product's going to be? 1,2, which is an alcohol. If I give you a alpha beta unsaturated ketone and a cuprate or some other nucleophile, you're going to predict that the major product is the result of Michael addition or 1,4 addition. That's right. Okay? So for example, cyanide is another nucleophile that likes to do Michael additions. Okay? So cyanide will react at the Michael carbon, break the weak pi bond, and give me the enolate intermediate, okay, which they're just showing the resonance structures here. And that is nice and stable until Maggie comes along and works up the reaction. And we know that that gets protonated, becomes the enol, which tautomerizes back to the ketone. Okay? Cool thing is she just formed a carbon-carbon bond at the Michael carbon. Okay? Amines also prefer to do Michael chemistry. What did we learn about in previous chapters? If I have a primary amine and a ketone, what did we get? Just a ketone by itself, what did we get? An imine. Not when we have an alpha beta unsaturated ketone. When we have an alpha beta unsaturated ketone, the primary amine, actually the secondary amine as well, will just act as a nucleophile on the Michael carbon and give me a new carbon-nitrogen bond on the beta carbon. It leaves the ketone alone. Okay? So all you have to remember is that ketones, or excuse me, alpha beta unsaturated ketones or aldehydes react with Grignard reagents and lithium reagents in a simple fashion, and everything else is Michael. It's not that hard. It really isn't. Okay? If you can memorize that. Enolates also react in a Michael fashion. Okay? So, if I have an enolate and I have an alpha beta unsaturated aldehyde, it will form a carbon-carbon bond at the beta carbon. Okay? You got to practice this. But you have everything you need. I could technically stop teaching at this point in the semester and tell you to go practice. I will not do that to you. But I have taught you everything you need to know to get through all this. I promise. It's just figuring out the little nuances now. Okay? We're going to pick up with the Robinson annulation on Friday. We're behind a little bit this semester. That's okay. We're going to have to do a little makeup here in the next, probably next week sometime. So I'll send out an email about when we could have an extra class meeting to try to get caught back up so you have everything that you need for the ACS standardized exam.